So we're continuing now that I've swapped out the plastic. And we're going to talk about the different types of cells, which we've done before. Is we did this in the kingdoms. There are 2 kingdoms that are prokaryotes and 4 kingdoms that are eukaryotes, where the cells are different. So this is a bit of a reminder with a little bit more detail. Prokaryotes, much smaller cells. They don't have separate internal chambers of membrane. Sometimes the membranes are folded inside and you get kind of chambers that way. But they don't have a nucleus, and they don't have a lot of the stuff we're going to look at in the next chapter that are little bubbles or little chambers of membrane inside. So that makes them different. They have just one chromosome, and it's in the form of a loop, like the O in loop. Just goes around and around and around and connects to itself. And there's just one of them. Their chromosome number is 1. And they are asexual reproducers, sort of. Most prokaryotes are capable of making a little bit of they'll take a stretch of their DNA, a stretch of gene code that's useful for them, and they'll make an extra copy and make a little loop out of it. 1, 2, 3, 4, sometimes a few more genes on it, as opposed to the hundreds of genes they have on their own, or thousands of genes that they have on their own chromosome. That's called a plasmid, and it's shareable. They can pass a plasmid with a useful allele onto other cells, not even necessarily bacterial cells. That's one of the ways that we get bacterial genes integrated into our DNA, is by picking them up through plasmids that invading bacteria have put into our cells that just wind up being, stuck into our own chromosomes. It isn't sexual reproduction, but it does allow for more variety. If you get a mutation in a bacterium that turns out to be very useful, a very useful allele variant, they can make copies and just share them out. That turns out to have been a really major problem for medicine. Now, when I was a kid, back in the horse and buggy days, not quite, but my doctor made house calls. And you'd go into the doctor, and he would check you over, and he'd go, "You got a virus. Can't really do anything about it. I'm going to give you a shot of penicillin." And you go, "Why are you giving me a shot of penicillin?" And the standard answer was, well, the virus can reduce your they didn't call it immunity, your resistance, they would call it. And so we were worried that you might pick up a bacterial infection while you're fighting the virus. Turns out not really to be the way it works. And I suspect they knew it wasn't the way it worked, but it was a long time ago. But an antibiotic will kill prokaryotes. It poisons chemistry that happens just in bacteria. It doesn't affect viruses because they work differently. Penicillin is an antibiotic. But they figured, what's it going to matter? You don't have a bacterial infection, so we're not curing that, but what's it going to matter if we give you a shot of antibiotic? What no one was aware of at the time, that we've already talked about, we'll talk about some more, we're full of bacteria ourselves. We have it on every surface of us. We have it on our skin. We have it in our digestive system. We even have bacteria that lives in the acid of our stomach. Not friendly bacteria there, but we got bacteria all over the place. And in fact, when you get a shot of antibiotics, the symptoms that you get as side effects usually are not the chemical working on your cells, it's the chemicals working on your friendly bacteria, and it messes your digestion up and does other kinds of things. It might affect your resistance because you have bacteria that help you fight diseases. Anyway, so you're exposing your microbiome, your own bacteria, to this antibiotic, and you're killing at least some of them. And you are changing conditions. Remember, evolution works through changing conditions? You're changing conditions that favor a mutant bacterium whose chemistry is different enough that the antibiotic, the penicillin, doesn't affect them. So you get a shot of penicillin, it doesn't hurt them. They're your friendly bacteria, what do you care? But now you've got a whole population, because they keep making copies. You have a whole population of penicillin-resistant bacteria living in you. And then later, if you get a bacterium that is possibly going to give you a disease, your bacteria are not very discreet. They'll go to a new invader bacterium and go, hey, buddy, nice to see you here. What's going on? You friendly? Nah, well, you want a plasmid? And they can transfer the allele that gives them resistance to penicillin to the invader. So the invader doesn't even have to evolve a resistance to penicillin. They picked it up from your buddies that have kind of betrayed you. And that's why if you go in and you have a viral infection, they don't give you a shot of antibiotics anymore, because they don't want to set that whole thing in motion. And it could be months between the shot of penicillin and the actual bacterial invader giving you problems later on. But eukaryotes, bigger cells, internal chambers separate from the membrane, pairs of chromosomes, so I'm not writing this down because it's just, kind of single versus pairs. And our chromosomes are, if I could pull my finger off, don't pull my finger, are two-ended. Our chromosomes have two ends to them as opposed to being a loop. So we have matching genes. It makes us capable of sexual reproduction, where we can split our sets apart, have one whole set of genes we pass on, and then mix it with another set. And even if we do that within our own cells, we'll get different combinations than we would otherwise. We've talked about that before. But eukaryotes don't make plasmids, is that they don't need to. They pick up a lot of variety by having this mixing of genes happening every time reproduction happens. I want to throw a term out there. Your chromosomes are all numbered. You got a number 5, it's got certain genes on it, make certain proteins. You have another 5 you got from your other parent. It's also got the same genes. Now the codes may not match exactly. There could be a different allele. But you have genes doing this, making the same sort of protein up and down the number 5 chromosome, up and down the number 9, up and down the X's if you have 2 X's. Is that the only thing that doesn't work for is in me, I've got an X with a bunch of genes on it, nothing to do with being female. And I've got a Y with genes on it that only have to do with making an embryo become male. So that the pairs get a little weirder with that kind of sex determination. It's not the only way to make males and females. We'll talk about that later. But it is one way to differentiate a male embryo from a female embryo, is with certain types of chromosome setups. And we've got these two-ended chromosomes and pairs, matching pairs of them. There are size limits on cells. So I'm going to move this and put this up. Now this is what they call a logarithmic scale. So every line is 10 times bigger than the line below it. So it's not 1, 1, 1, 1. It's by 10, and then 10 times, and then 10 times, and then 10 times. And it's just changing the units from nanometers to micrometers to millimeters to centimeters, because the metric system is also a base 10 thing. So you can see here's a person. You can see stuff that you can actually see with your eye. Here you use a light microscope. Here you use an electron microscope. There are electron microscopes that can see atoms. There's pictures that your book links to. They don't look like much. They look like bumps, but because you're just looking at the outside of the electron clouds. But it's kind of cool that you can see an atom through a microscope. And this is the zone that cells typically are in. So they don't get smaller than this. Viruses are smaller than that. when it says smallest bacteria here, there are some bacteria, they are very, very small, but they live inside other cells. And so they let the cell around them do a lot of the chemistry. That's the only way they, problem with getting small is you need room to do stuff. And there's a certain limit where you get too small, you can't do everything you need to do. So you need to be in something that will do a lot of that stuff for you. Remember, that's what viruses do too. They go into the cell and take over the cell's production machinery to make more viruses. They don't have the chemistry to do that on their own. But here's the top edge. Their eggs don't really count. You'll hear like, what about frog eggs and chicken eggs? They're not really one cell. So they're up here, but they're not really single cells. This is the range, and you don't get cells that are big. Now the classic explanation for that, I'm going to tell it to you, but it doesn't make any sense. If you were producing bottles and you had to switch off your machinery from making 1 liter bottles to 2 liter bottles, those 2 liter bottles are not going to use twice as much plastic as the 1 liter bottles do. Twice as much volume, 2 liters, 1 liter, twice as much volume, but it doesn't use twice as much plastic because when you double the volume, you don't double the surface. So the bigger a cell gets, the more juice it has in it, the more stuff it needs to do with that juice, the slower the outside surface increases. It's actually an inverse cube law. You don't need to know that. So the surface can't keep up with the increase of stuff going on in the cell. That's the classic explanation. And it works fine if every single cell in the world was shaped like a soccer ball. Is that it's a math relationship for spheres. Doesn't even work exactly the math way with bottles, because bottles are long and oval. Some of our cells are completely flat. You'll see that when you look at your cheek cells through the microscope. They're flat. You double the size of a cheek cell, you double the volume, the surface pretty close to doubles too, because it's very thin. So if you expand the volume, you tend to make the thing just bigger and still flat. So this explanation for volume and surface doesn't really apply to an awful lot of cells in the world, so it doesn't really make sense to say that's why you don't get cells above a certain size. So I'm going to throw my idea out here, is that I'm not alone in thinking this, but it isn't an official theory anywhere. What you find is cells get bigger and more complex. Remember single-celled, multi-celled, colonial single-celled in between? That it is more efficient to not just have chambers inside the cells, but actually separate into separate cells that are doing different things, that are all aggregates together, like they were a multicellular system. And that's more efficient than a big cell trying to do everything at once. And I do believe that the reason you don't get big cells is that anytime there was a competition between a single cell colonial and a big cell trying to do the same thing, the single cell colonials were just better at it, and they outcompeted the big cells. You can find some really big cells in the world. If you want to go like 5 miles down in the Marianas Trench in the Pacific, deep, 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 high pressure water, not a lot of living things there, not a lot of competition. They have discovered amoebas that you could actually see under the without a microscope, big old guys. But even then, not huge, probably in this range of size up there. So that's the idea of cell size limits. And we're getting close to the termination edge for the second exam. We're not quite there yet, but we will be soon.